session on day 4 of AICTE sponsored STTP 2021. We are fortunate to have Professor Umesh Patil as our guest speaker for this session. Before starting for the lecture, I would like to introduce the speaker to our participants. Professor Umesh Patil is presently working as Professor of Pharmacognosy, Dr. H.S. Gour Vishwavidyalaya, Sagar, MP, and serving as General Secretary to Indian Society of Pharmacognosy. Sir has completed his B.Pharm, M.Pharm, and PhD from Dr. H.S. Gour University, Sagar, and postdoctoral studies from Institute of Biology, Leiden University, the Netherlands. Professor Umesh Patil has more than 22 years of teaching and research experience. Adding to his credit, Sir is recipient of 7 prestigious awards given in the field of HMPs. Sir has published 110 papers in reputed journals and has been serving as editorial board member of many reputed journals. We welcome you, sir. With these words, I request Sir to deliver his deliberation. Please, sir.
Thank you. Thank you, Ruchi ma'am. And I am grateful to Dr. Shiv Shankar Shukla and all the fraternity of Columbia Institute of Pharmacy, Raipur. My special gratitude to Mr. Hura. I don't know he is here in this meet or no, but I'm grateful to all of you. And before I start, may I request all the participants, can you put on your camera? I just wanted to see you on the screen, if it is possible.
All the participants are requested to kindly turn on your camera.
See, we are teachers and it's a STTP. So before delivering, I love to see you. That's it. That's my intention. And then later on, you can put off. Thank you. So my topic is, see, whenever we talk about anything, it must be like a two-way kind of discussion. So I believe that whenever we produce some information or we transmit or impart some information, there must be some interactive kind of discussion. I believe this is my opinion. And whenever you have some queries during discussion, since my talk is based on natural products and herbal medicinal products available in market and challenges which are associated for developing various kind of herbal products, not only for cosmetics but also as nutrients and functional foods. And that is why I chose my topic to have the challenges which are not only for the production in different pharmaceutical company, but it's always all the challenges are dependent on what particular kind of material one company gets from supplier. So starting from the cultivation, collection, processing, transportation, and all that, we have to look into the challenges which are associated to such type of the products. So my talk is, as I told you, phytomedicines and the challenges and research opportunities. Is it visible to you, my screen?
Yes, sir. Yes, sir. It's visible.
Okay. See, phytomedicines is a very vast area of the therapeutic agents which comes from various natural sources, but they are having inputs from some botanical metabolites as well. And that is why secondary metabolites from botanical sources, they have their own importance. Because whenever these secondary metabolites are biosynthesized or developed inside the plant material, they have a very long journey to develop themselves as a molecule of therapeutic importance. During their development journey, different kind of precursors, some intermediates, and various kind of biochemicals are involved. So altogether, we can say that whenever we have one active molecule, maybe in terms of some alkaloid, glycoside, or any other kind of secondary metabolites, we have one past history of its development. Likewise, when the particular molecule of importance is isolated, when it is separated from its different kind of the joint molecules or complex kind of attachments, then this isolate is having its own stability, its own life, its own toxicity and safety. So altogether, when we go for the development of phytomedicinal products, we have to look into the challenges which are associated to the development of the particular plant product and all the factors which involved from plant to the patient, like from farmland, from agricultural land, or from wild sources, from forest area, or from any other sources where botanical has been developing, from that particular side to the patient part, to the hospitalized one, or outpatient or inpatient, or to the user or end user of that particular product. We have to look into all the challenges which are associated to these products. And that is why my talk will lead you like different type of challenges. So when we go for the medicinal product of herbal origin or phytomedicine, traditionally we see that these are the products which have the therapeutic efficacy and least or very less number of the undesirable effect, which is frequently quoted in many texts. But unfortunately, this kind of statement is not justified statement, because when we say, if one statement says that the herbal products are less toxic, they are always safe, they have no side effect, this statement is misleading statement. And that is why being a teacher, we have to justify that. We have to aware our students and researchers that the product of botanical source or herbal source, they are also equally responsible for some untoward or undesirable effects whenever they are consumed in terms of pharmaceutical product or herbal medicines. So for the challenges are concerned, say the first challenge, that is the less than desirable quality. Why this challenge? Because because of misleading information, because of misleading kind of the claim which has been labeled on the particular product, whether it is semi-finished product, extract, or some supplied crude drug, the supplier is claiming it's a overall activity beyond its natural importance of the therapeutics, and that is why this misleading information is evolved, and that is why it is considered that the product which was supplied by the supplier is having less than desirable quality. In fact, that quality was it's a natural kind of phenomena, that the quality is dependent on secondary metabolite. That's it. If secondary metabolite is dealt in a proper way, if it is quantified, if it is characterized, if it is assessed in proper way with the involvement of the optimum number of the analytical techniques which are available till date, then definitely the claim which will be labeled on that particular product won't lead like less than desirable quality. And that is why for us, for teachers and for the researchers, we have to consider and we have to pay our attention, optimum attention, and our attention should be like that what particular range of the actual concentration and quality is present in our product, it must be claimed accordingly. And that is why less than desirable quality is one of the challenge. Then the next one is like single target, single compound, or one disease, one drug.
Sir, sorry to interrupt you, but your slides are not rotating.
Okay, my slide is first slide. Is it not visible? Challenges.
So yes, the phytomedicine challenges and research opportunity. The first slide is visible, but slides are not changing.
But I have just changed second slide. In my screen, I am seeing my second slide.
So, but it's your here.
Okay.
It's your first slide is only visible.
Okay. Now is it second? No.
No, sir.
Okay. So there may be something wrong or
Yes,
some.
sir.
Huh?
Sir. Yes, sir. Now it's visible. Second slide, challenges.
Okay. So just interrupt me whenever the slide is not moving, right? I am on
Okay, sir.
slide number 2, right? And discussing
Yes,
about
sir.
the challenges. So just now I told you, like we have less than desirable quality, and this quality related challenges based on the insufficient information, which are misleading sometimes to end users, to the pharmaceutical company, and to the researchers as well. And that is why we have to consider the overall quality of the secondary metabolites in terms of the its quantification and assessment with the involvement of modern analytical techniques. So that's the point I wanted to clarify, that this less than desirable quality is because of the reports and documents which are available in different segments of the either in research papers, sometime in some leaflets, and sometime on virtual kind of document and websites. So likewise, this misleading kind of desirable quality and less than desirable quality is because of ourselves. And the second one is single target, single compound, or one disease, one drug. See, for example, the herbal medicine or phytomedicine, there are huge opportunities to work on them. Why? Because the world is moving towards different type of sterilization with molecular mechanism, right? When we develop one sterilization procedure with the development of molecular mechanism of that product, definitely we have to look into wholesome quality of that particular product. So when we consider one extract or one particular active fraction that is containing more than some 10 or 20 components and phytoliths, which may have sometime one activity, say for example anti-diabetic, but at the same time some components may have antioxidant and other kind of therapeutic responsibility as well. And that is why the phytomedicines are having huge opportunity to consider, like sometime if single target is, we have single target, then we may consider it as a single compound, as a biomarker or chemical marker. Or if we need one disorder, then definitely one drug is sufficient. So likewise, challenges are associated in what way we have to move ahead for the research opportunity and the development of the phytomedicine. Then the second one is multiple target with multiple active principles. And in this way, our target is to develop some polyherbal formulation or some poly component which may come from different extract of either one drug or one herbal source, or sometime we may consider more than one herbal material for the development of the drugs for multiple targets. Then we know that the holistic approach is one of the important challenge, because the many developing many research organizations in developing world now they are interested for the active fraction instead of isolated one. The reason behind is because the overall synergistic responsibility and synergistic effect of the herbal medicine is because of the holistic kind of the performance of the secondary metabolites. I will explain you in what way this holistic approach is more important than the isolated one. For standardization and for the quantification of the particular quality control targets, we can consider one or limited number of the chemical markers in our product. But for the therapeutics, the holistic approach is popularly accepted throughout the world. And that is why under this holistic approach, the overall quality is maintained in such a way that the synergy of overall extract is maintained. And that is why we have to consider wholesome activity of the wholesome product. And that is why holistic is the approach which is based on all togetherness. So instead of putting one constituent, one isolated one, we have to consider for use as a one active fraction. Is it clear? Is it audible to you?
Yes, sir.
Okay. Then the next challenge is ethnomedicinal input and its application in modern herbal medicinal products. See, ethnomedicinal is one of the challenge. Sometimes some people consider that it's area of anthropology or it's the area of some ethnobotany, and it's not related to pharmaceutical products and all that. But ethnomedicinal inputs are very, very important because see, we know that traditional system of medication that is based on traditional inputs, traditional information. But over a period of time, maybe after one decade or maybe after 100 years, the information and the valuable infos which are based on the consumption and the production of the extract, they are getting deviated from its original information, and that is why sometimes it is said that the decoction is the more active than the investigator. They look into the hot extraction, and sometimes they just incorporate some alcohol content to get more extract and all that. But because of the exposure to various solvents, there are chances of the degradation of certain kind of the secondary metabolites, and because of that this particular ethnomedicinal inputs are getting changed. This is one way. Another one is like when we collect the medicinal plants material from either farmland, agricultural land, or from wild sources. The way of picking up the plant material, it has its own importance. Likewise, we know that the time of collection, the way of the the storage of the collected material, the way of the packaging of the material which was collected from farmland, and transportation from that site to the laboratory condition or the production house, are all together they affect the overall quality of the wholesome activity of the product. And that is why ethnomedicinal inputs have their own importance for the development of modern herbal medicinal products. Then another challenge is like chemo varieties. We know that the chemo varieties are getting changed from the point where we harvest the material and the point where we use the material in the laboratory or production houses. The reason behind is various kind of exposure of environmental factor. There are the factors which are underground factors and on the ground factors. Like during agricultural practices or good agricultural practices, we are not in position to control various kind of factors which are coming maybe from water side or from the various kind of canals and various kind of soil qualities and all that, because we know that when this biosynthetic pathway is ongoing, there are chances of the exposure of that biosynthetic pathway or that exposure of root part to various kind of toxins which are coming from the underground sites. And likewise, on the ground factors like if the cultivation and the production of that plant material is in the area which is adjoining to various industrial setup, and there are some different type of radioactive sources in the area adjoining to that particular farmland, then definitely the biosynthetic pathway will be affected by these factors. So these are the factors which are on the ground factor which affects the overall biosynthetic pathway, and that is why chemo varieties exist. So we can't say that 1 year back we have used one species from the same site of production will lead us to next year in same manner, because we don't know that in what way the different kind of chemo varieties may exist inside the plant. And that is why it must be checked with the help of various analytical techniques in appropriate way. I'll be discussing later on in what way these kind of challenges are dealt with. Then the source and the quality of raw material are variable always. So various, this is one of the area of, again, the research opportunities for the, supply chain of the material, which are based on the constituent variability and sometimes quality variability, which is to be, considered into, regulatory norms and all that, and that is why it remains one of the challenge for the phytomedicinal investigators. Then sustainable growth and conservation of plant species. We know that many species and many plant species, they are not available abundantly throughout the world, and that is why one species sometimes they are getting endangered from one location, and sometimes it is getting evolved in another geographical location. The reason behind is harvesting and uncontrolled harvesting of the plant material, and sometimes maybe because of weather condition, the plant is getting endangered, and that is why we have to pay the sufficient attention for the sustainable availability and sustainable growth of that plant species. Then another challenge is herbal products, which are usually mixture of many constituents. So to develop the overall profile of the product, we have to look into various analytical techniques, and fingerprinting is one of the most common technique. And these days we use various kind of molecular techniques by which the overall profile of the mixture with many constituents may be developed in one plane, and ultimately that may be considered as a reference kind of profile for the comparison in future. Then another challenge is the active principles are unknown in many cases, and which remains one of the expensive effort for the investigator. That is why this remains one of the challenge for the standardization purpose. Then selective analytical method and reference compound may not be available always, and that is why the wholesome profiling in terms of fingerprinting is important, because we don't know what are the number of reference compound which are present in our test sample, and of course test sample may also having selectivity of the analytical methods because of the n number of factors which comes from the secondary metabolites. So altogether, these are the challenges by which we can consider that the phytomedicines are having their huge research opportunities for the purpose of either to develop finished formulation, semi-finished formulation, or to develop some standardized extract. One opportunity which we all have seen during past few months, like COVID-19 and herbal product. Sometimes there are reports that Ayurveda and the text of Ayurveda and some inputs of Ayurveda are also explained in big way for the group and diversified severity of this particular COVID patients. So likewise, we have seen that the various kind of immunostimulant products are available, and of course in India it was consumed by many people during past 1 year for the purpose of the immunostimulant. So this is how this slide is depicting, like different type of approaches may be evolved with the help of various medicinal plants. So this is one of the opportunity which we can consider for our research programs. Importance of medicinal and aromatic plants with reference to Asian region. We know that Asian region has abundant species of medicinal and aromatic plants, and traditional medicine has been practiced in Asia since very old times. And only two region, India and China, they have made use of medicinal plants to cure many ailments for thousands of years. And the concept of phytomedicine was evolved in this region also, in this region only. And that is why the contribution of China and contribution of India, especially in medicinal plants, is recorded as a distinguished contribution to the area of phytomedicine. And we know that according to WHO, the goal of health for all cannot be achieved without herbal medicine, because in developing world, nearly 80% of the population is dependent on such type of product. See here, I want to emphasize one misleading information in many texts, that the whole world, in many books and reports, it is mentioned that 85 or more than 85% of the population of the world is dependent on medicinal, herbal medicinal product. That is not true. That is developing world, not the whole world. 85% or 80 to 85% of the developing world population is dependent on such type of product. Then importance of this product with reference to our country, because we know that the international market of the herbal product is, which is estimated to be more than 60 billions, and which is poised to grow 5 trillion by the year 2050. And this is the one expectation which indicates that the production, consumption, and domestic and international trade in medicinal plant-based product is going to grow at very significant growth rate. The sector of medicinal plants and aromatic plants in India has several strengths. We have enormous biodiversity. Then we have different type of soil conditions, different kind of climate condition, very rich heritage of traditional system of medicine, a strong base of research and development laboratories. Then we have skilled manpower, lower production, then manpower cost. Yes.
So it's not changing again. So kindly, yes, click your slides from the left side.
From the left side.
Yes, sir. So it's on second slide, challenges.
Okay. So better to go again like
yes,
this.
Sir. Yes, sir.
Should I, exit from display mode?
So you can do it from your entire screen. Share it from your entire screen, not from window.
Okay.
Yes.
Okay. When I share it from my screen, then it will be visible.
Yes, sir.
Okay. Just a moment. Should I stop first?
Yes, sir. You stop your presenting, stop and share it again. Yes, sir. And yes, sir, entire screen.
Okay. Just a moment. Is it visible now? No?
No, sir.
Just a moment. Share. No, but I am trying to, present entire screen.
Present. Click your screen button.
Then
and then entire screen. Sorry, Windows.
Window, na? Okay.
Yes, sir.
Because when I go for the entire screen, it's not coming.
Yes, sir.
For window, it is coming now. But I'm trying to. But is it visible now?
Sir, it's visible now. Important. Kindly change your other slides also. Yes, sir. Now it's visible. Strength of the.
Okay. If I go for full screen, then it's okay.
Yes, sir.
Okay. So I was discussing about the strength of this sector in India. Like we have enormous biodiversity, which is very important. See, I experienced when I was working in one of the laboratory in Europe. We were working to develop metabolic profile, that is metabolomics. During those time, we were not able to collect the sample from the biodiversity regions, and that is why we have to travel to the Asian region. Our researchers, they have collected different samples from different countries. And for such type of activities, we know that it's expensive, and the collection is also one tedious job to get it collected in different countries with clearance of their immigration requirement and all that. But in India, we know that we have huge biodiversity. We just collect one sample from Himachal Pradesh, then from Bhuj area, or from West Bengal, or from the Kanyakumari and the Tamil Nadu region, then definitely we will be having the biodiversity kind of opportunities. So likewise, the India is having different type of positive points for the phytomedicinal research. Then that is why I would say that the bioprospecting in India is very, very important, and it's more feasible than in any other region of the world. The reason behind is we have the abundant kind of different type of weather conditions, and different type of biospheres are available in one country only. You can travel throughout the country and collect the plant material, and you can involve your whole research group in the bioprospecting of these medicinal plants. And we know that bioprospecting is directly related to IPR issues and all that. So the country is very rich in terms of the various kind of reserve forest and reserve locations for the purpose of the this plant genomics and other type of research targets. See, as far as the India is concerned, is it changing now, Ruchi ma'am?
No, sir. Sir,
Again?
yes, sir. Yes, and now it's changing. You can use your cursor.
So I will keep on there
from your left side.
Okay.
Yes, sir.
So this is how I have depicted here that we have huge very strong resource base where nearly 45,000 plant species are there in our country. Then 3,700 species, both higher and lower plant, are of their therapeutic values. 1,000 medicinal plant species used by various contemporary industries of Indian origin and Indian side, and 80% are produced from wild areas, mostly notified as a forest land, and only very limited proportion, that is 10 to 15% medicinal plants are produced in cultivated and farmlands. It means wild area is contributing in big way, and the medicinal plants are available in various forest sites, and that is why the overall biosynthetic pathway is not deviated from its natural one. Because when we go for the medicinal plant production in a particular farmland, there are chances that the chemo variety may be getting deviated from its biosynthetic pathway. Then the forest of, we know that Himachal Pradesh and the Western Ghat. These are the two very rich sites and a very large proportion of medicinal plants which are available in India. One is Himachal Pradesh and Western Ghat. Have you heard this Western Ghat? Anyone can? May I ask to someone that, have you heard this Western Ghat? Lubhan Singh. Mr. Lubhan Singh is there in audience? Dr. Gayatri, S. Gayatri, Faculty of Pharmacy. May I ask Dr. Prashant Tiwari if he is there? Shiv Shankar Shukla ji. Dr. Shiv Shankar Shukla. Are you there? My voice is audible. Anyone can respond?
Yes, sir, you're audible.
But Ruchi ma'am, where are the participants? My
Uh,
voice is not reaching to them.
no, sir. It's
Yes, sir, it's audible.
It's audible. Okay. So I will just now talk to Mrs. T. Sheila Rani. Madam, have you heard about Western Ghat?
Yes, sir.
Western Ghat.
Yes, sir.
Where is it? Is can you throw some light on this Western Ghat biodiversity? Can anyone tell us about this Western Ghat region? Anyone, please. आप में से कोई बता सकता है वेस्टर्न घाट के बारे में थोड़ा? सचिन, मिस्टर सचिन जैन, IPA. रुचि मैडम,
yes, sir.
I guess participants are not responding. There are certain problem. I think they are not listening the voice, or I don't know what's the reason.
Sir, but your voice is very much clear.
It's clear to you, but not to them, I think. Is it so?
Your voice is clear, and your when your slides are also seen.
सब दिख रहा है ना, बट मैं किसी से पूछना चाह रहा हूँ, इंटरेक्ट करना चाह रहा हूँ, बट नो वन इज रिप्लाइंग. वैभव त्रिपाठी.
Yes, sir.
Is it audible to you?
Yes, sir.
Good. So have you listened about this Western Ghat I was talking about?
Western Ghat is located west coast of India. West coast of India.
Yes. So this, this Western Ghat is the one of the biodiversity region, which is considered as the largest producing geographical location of medicinal plants in our country. Okay. Right. So based on this particular, some plus points which are associated to our country, we have different type of 5 opportunities. So my topic was like phytomedicine and research opportunities, and now I am discussing about what are the 5 opportunities which are associated with phytomedicine. Is it audible to you, Vaibhav?
Yes, sir.
Okay.
Yes, sir. Audible.
So the quality of the medicinal plants, when we consider about the various parameters related to quality, we see that there are 5 opportunities which are available for us to go ahead. Like one is global business angle. If we are able to develop the quality medicinal plant material, either through drug or the extract or the semi-finished material, then definitely we will have the different agencies worldwide which are engaged in the business of such type of products, and definitely if we will be able to supply the quality material, the material will be accepted throughout the world. Then the next one is the quality with safety and efficacy issues. So when we consider the quality of the plant material, definitely the safety and efficacy issues will be addressed in big way. Then the third one is the overall development of phytoleads. Phytoleads are nothing but when the constituents which are present in our product, in a plant product, that is considered as a leader, maybe in terms of therapeutic leader, or maybe sterilization leader or quality control leader. So what we do here, we develop certain type of reference components which are used for the purpose of the routine kind of sterilization and routine kind of checking of the product by considering the quality of the phytoliths which is present in the supplied material. So phytoliths are also itself is having one global opportunity for to develop it, characterize it, and sell it. Then current therapeutic priorities. We know that this is the era of communicable disease. Before some time, non-communicable disease was was the prime goal of the different health agencies, but these days communicable diseases are also having the equal kind of challenges which are associated to develop therapeutics. So current therapeutic priorities like antiviral drugs and different type of other kind of therapeutics definitely have the opportunities for the phytomedicinal investigators and herbal practitioners. Then the fifth one is the veterinary area, where various herbal medicinal products, they are developed for the purpose of animal health care. So this animal health care itself is having its a vast opportunities and huge scope of the import and export of the product. So in this way, we have five global opportunities for the purpose of development of various herbal material. When we talk about the global business and the research opportunities, we see that the overall options we have in terms of the medicinal plants and medicinal herbal products, like we can go for phytopharmaceuticals, then the development of modern drugs, then we may go also for different type of drug leads, then different type of drug intermediates, then different type of preparations like herbal drug preparations, then different type of traditional medicine, complementary medicine, household remedies, then nutraceuticals, functional foods, then food supplements, extracts. Then if we look into different type of essential oils, various kind of flavor industry, fragrance industry, then perfumes, various kind of cosmeceuticals, then entomological products like various kind of insect repellents, insecticides, pesticides, fungicides, and in addition to all these therapeutic agents, different kind of pharmaceutical aids, different kind of the diluents, gums, mucilages, and various kind of sweeteners. So altogether, there is a huge global business opportunities, and the research opportunities are existing in this particular sector. So this way, the one can choose any particular area of investigation and go ahead for the purpose of the development of the phytomedicine. As far as this Indian herbal exports is concerned, you can see that the export value of herbal production from India to is during the period 2015 to 2019 is nearly about 500 million US dollars, and it was a little bit setback during the past 1 year. Otherwise, it is going up and up. And the reason behind is the requirement of the basic requirement of the product that is now being fulfilled in terms of safety and effectiveness. Why? Because we know that when we practice the different kind of traditional medication system, we have to justify the safety and the effectivity, and both the things can be justified on the basis of secondary metabolites. So the overall quality of the product is safety and effectivity, and the quality of the product will be grouped in modern era of science with the help of external and internal. There are two type of quality we have to look into this product botanicals. One is external, which is including various kind of factors like toxic metals, then opportunistic type of incorporation of various residues like pesticide residues, microbes, sometimes some intentional type of external issues like adulteration and misidentification of the drug is done. So likewise, various kind of quality issues are associated with the product, and internal issues like there is complexity and non-uniformity of ingredients in herbal products are the internal parameters which affects the overall quality of the phytomedicinal product. See, when we go for the overall movement of our product, it is being affected by a number of factors and locations. Starting from cultivation and collection of medicinal plant, we know that sometimes the plant material directly comes to market, and from market it reaches to consumer. But in many times it is traveling through dealers, suppliers, then various kind of processing factories, and from processing factories it reaches to processed herbal materials. From this processed herbal material, it is reaching to market sometime and going to consumers, or sometime it is reaching to pharmaceutical production houses. Then again from production houses it comes out to the dealers side, then clinical hospitals, and from this hospital it reaches to practitioner or herbal practitioner and to the consumer. So the purpose to show this particular depiction is how the medicinal plant product is, or herbal product is getting exposed to various locations and sites and number of factors from where it is being affected in terms of its safety and quality. So in this way, the various kind of quality challenges are getting involved in the process of the travel of the transportation path of the herbal medicine product. Is it getting changed, Ruchi?
Yes, sir.
Okay. So now I'm showing you like there are different success stories of the phytomedicinal opportunities and research opportunities which have revealed various kind of businesses, like during past few decades different type of artether antimalarial molecules are available throughout the world in hospitals, which was obtained from the species of the Artemisia like Anuva, Afra, and other kind of Artemisia species. Then we know that colchicine, which is being recognized as a very prominent kind of antidote option for the therapeutics. Then digoxin, morphine is, we know that it is having its own importance. Various kind of galantamines and different type of ephedrine and vincristine, vinblastine for the different kind of anti-cancer regimens. So these phytoleads are having its own importance, and likewise there are huge scope to develop these phytoleads, and of course it's expensive and time consuming, but once we start working on one particular medicinal plant, after a few years, maybe 10 or 15 years, we will be able to give one individual molecule of importance which may be recognized globally. When we go for medicinal plant research, our aim is to develop certain kind of parameters which are considered as standard parameters, and that is why standard parameters are leads us to the quality control domain. So standardization is the process of developing and agreeing upon some certain technical standards, and we know that these days regulatory agencies they keep themselves on dynamic kind of challenges. Like yeah, now sterilization is not a specific kind of sterilization. Every moment of time, the whole process is getting evolved, improved, and getting changed. And time to time, various regulatory agencies, they are also adopting the newer techniques and novel analytical tools for the evaluation of these herbal products. So sometime we have heard that HPTLC was not the official process of analysis. When during the 60s and during 50s, but these days many regulatory agencies, they have included HPTLC as one of the tool of quality control process, so that we have to develop the overall profile of that particular test sample in terms of its planar profile or chromatographic profile, which was developed with the help of planar chromatography or HPTLC. We at our council, that is International Council for Medicinal and Aromatic Plants, we develop different type of dynamic upside attributes. Upside attributes are nothing but these are the attributes which are responsible for the authenticity, purity, safety, and assay. So in what way one particular regulatory agency is putting norms and regulation for the authenticity, it has its own challenges, like in what way gross morphology will be decided, in what way microscopy will be completed, in what way chemical analysis will be developed, in what way the fingerprinting or DNA fingerprinting is developed and reported. So altogether, these are the techniques by which the overall identity and authenticity is confirmed. Then purity is nothing but as per regulation, we have to ensure that there is no adulterant. But the claims regarding no adulterant must be developed with the help of modern analytical technique. You can't say that by simply spectroscopic method you say that your product is not having any adulterant, or it's a very pure, or purity is checked with the help of the available method of analysis. It should not be available method. It must be as per the regulatory methods, regulatory procedures. Under APSA, safety is to prove its safety and quantification of undesirable and harmful effects. So as far as the various secondary metabolites are concerned or contained in the product, we have to evaluate the product to explain how the undesirable and harmful effects are associated with the supplied material. Then assay, again, this assay procedure should have the capability and must have capable enough to develop a chemical and biological profile which could assess the chemical effects and overall therapeutic values. So in this way, upside attributes are always remains dynamic in terms of the its execution. And as per ICMAP guidelines, we go for different type of involvement and incorporation of latest technology and modern techniques of analysis. These are some parameters which are actually general parameters and conventional parameters, but the development of the protocol should be modern development, like general data collection, the way of geographical data harvesting time, processing time. It must have real-time recording. It must have the recording technique which records the real-time documentation. Then identity, it must have a different type of molecular fingerprints. It must have the modern kind of fingerprints which are associated with the original one. Then, as I told you, contaminants and the purity should be checked with the help of modern analytical molecular techniques by which we can decide that the overall profile of aflatoxin and different type of other undesirable content which may incorporated during the path of transportation of the herbal material. So altogether, this upside attributes, they are executed, they are getting checked with the help of some important analytical tools, And these tools are why important, because we have to prove constant composition of the supplied material, and educate involvement of the techniques like spectroscopic analysis. Then it must have hyphenated techniques with like TLC, HPTLC, HPLC, GLC, sometimes some pressure chromatography, then molecular fingerprinting, then chemometrics. We know that chemometrics is, I will explain you in what way the chemometrics is more important for the purpose of herbal medicinal plants, because herbal medicinal products, because chemometrics is the technique by which we superimpose various kind of data, and ultimately with the help of that particular comparison and correlation regression, we will be able to reveal certain kind of the quality explain and quality kind of claims on the basis of this regression and correlation. Then biopharmaceutical characterization, metabolomics, molecular imaging, and molecular mass imaging. These are some tools, analytical, modern tools, which are involved for the purpose of analytical domain of herbal medicinal products. These are the opportunities by which we have to go ahead for the development of our research protocols. Is it audible to you? Analytical procedure, Ruchi?
Yes, sir.
Okay. So I won't be going in detail with like fingerprint and all that, but my intention is to, when we go for the herbal medicinal research, we have to involve the analytical tools which are as per the requirement of the council, as per the requirement of International Council for Medicinal and Aromatic Plants. See, when we go for different type of this modern techniques of analysis, some months back, like in 2000 and also in 2019, the reports of the adult rains were evaluated and evaluated by various regulatory agencies on the basis of fingerprints. They have evolved fingerprint techniques in their official process, and ultimately on the basis of fingerprinting, they have compared the different products, and they have found, you can see in this picture, that the percentage of the various kind of authentic product and spurious and substandard products. So likewise, on the basis of fingerprint, we can say that this is one of the very, very important technique for the purpose of the adulteration. Chemometrics, as I told you, it's a kind of interpretation of three-dimensional data, where we go for two principal component. What we do in this two principal component, we compare the data set which is coming from one side, like cultivation side, then another one is from biosynthetic pathway, then another one is from harvesting side, and ultimately the all data is compared with the data which is present at the time of the processing of the raw material. So all together we are able to find out in what way the constituent variability is getting evolved in the product. In this way, a different type of chemometric data is getting evaluated, where different fingerprints and different type of sorting of the information is done, by which marker compound analysis and herbal product fingerprints with multi-constituent determination can be achieved. So multi-constituent determination is essential for the purpose of it's a synergistic response, because we know that when the one particular biological activity is because of altogether efforts of many secondary metabolites, and when we evaluate the overall bioactivity with the help of such type of comparative chemometrics, then definitely we'll be able to develop the overall synergistic response. I work in metabolomics, and definitely metabolomics is one of the important techniques. So I will throw some light on some information on the metabolomics. Ruchi madam, how much time left for me?
So you can use 5 to 10 minutes.
Okay, so I will go a little bit quick way. See, metabolomics is since I'm working in this techniques for last 20 years, and we have developed many kind of profiles of medicinal plants, and you know that in European world and the US, metabolomics is one of the important technique by which the botanical products are getting justified for their safety and their effectivity. So metabolomics is nothing but it's a kind of holistic approach. As I told you, plant to patient platform means when we develop one plant, starting from that particular point of time to the end point, that is the site of the end user, what is going on with the plant material, that is plant to patient platform. What we do in metabolomics, metabolomics is since I told you it's a profiling of wholesome, right? Wholesome of the plant, whatever plant is having, whether that is primary or secondary metabolite, whether it is intermediate, whether it is inactive or phytoleads, whether it is biomarker or chemical marker. All together we have to develop their role as and when they are involved in the particular pharmaceutical product. So what we do, we collect the living organism from the plant site or the different production site. Then we go for sampling, where we develop some sugar sample, liquid sample, secondary metabolites, some amino acids, and with the help of some organic acid, we go for its derivatization. So during derivatization, different type of complexes are formed, and these complexes are subjected for analysis to develop the spectra using various kind of molecular techniques and different type of spectrophotometric techniques, where we develop huge number of data sets, and this particular huge number of data sets will represent all together holistic kind of nature of that particular product. Then we go for the data conversion. So as per our needs, we convert all the data set for the requirement, and then we go for the data mining. So as per our needs, we just reduce the number of data. So we just try to ignore the data which is not for the purpose of our functional elucidation, and which is not related to our gene function. And ultimately what we do, we just shorting our desirable data set, and ultimately we'll be able to develop one functional elucidation for a particular gene function towards therapeutic importance. So ultimately, at the last, the metabolomics is having its capability to give the feedback to the bioscience for the purpose of safety and efficacy. So in what happens in metabolomics, when we develop the metabolomics, as I told you, it's a metabolome, so wholesome. So metabolome is representation of either biofluid, some metabolic footprint. Means when one biosynthetic pathway is ongoing, always, every time, every point of time, it is revealing some footprints. So all together, when we are able to characterize, recognize the footprint, then definitely it will contribute for the metabolome profiling. Then in addition to biofluids and metabolomic footprint, it also having the characteristic of intracellular metabolites and the characteristics of various tissues. So altogether, this metabolome can be developed with the help of various analytical techniques, and commonly used techniques like GCMS, LCMS, FTIR, or direct injection mass spectroscopy, NMR, or other type of mass imaging microscope is also nowadays available, by which immediately put your sample and it will give different type of mass peaks, and ultimately you will be able to find out in what way different type of mass is present. This is the multi-dimensional kind of scope of metabolomics, which is depicted here in this slide. In our council, we have various kind of initiatives like knowledge-based drug discovery from natural sources, where we use to develop multidisciplinary approaches for drug discovery from the natural sources. We have developed many projects like field-based studies, which is having the different type of plant collection targets, then processing of the plant material, then whatever material is collected from various locations, they are transported, they are brought to the laboratory condition, and ultimately laboratory conditions are one of the option where we can test our various kind of available efforts for the development of quality control parameters. As I told you that fractionation is not desirable because esterification is one of the process which involves in the process of fractionation, by which sometimes phenolic substances are getting oxidated. So by this oxidation, energy can also be challenged. Then we know that phyto research is having its multi-dimensional therapeutic tools. Like in first slide, I told you, like if our target is wound healing, we know that wound healing is not a single process. Wound healing is the process of mixture of processes like cell proliferation, collagen formation, then contraction of collagen lattice, then microbial infection, then destruction of cell and tissue by reactive oxygen species. So altogether, different concentration and the quality of secondary metabolites and different phytoliths, they have different kind of responsibility. So altogether, some phytoliths may have their effect on proliferation. Some of them may contribute for collagen lattice formation. So altogether, when we go for holistic way, by involvement of holistic way, definitely we will be having opportunity to develop the synergistic response. Then in 5 minutes I will take you to the medicinal plants and phytomedicine, which are linking the doctrine of signature to human health. Like when we go for the new strategies for the research for the involving medicinal plant, definitely we may consider some doctrine of signature. Means what kind of symbols, what kind of the characters, colors, and different type of structures plant having may have contribute for the same type of the therapeutic goals. Like if you see here, this is frequently quoted that the carrots, or traditionally it was claimed that carrots are having its activity on this performance of eyes. Why? Because there is the resemblance of the structure on the transection of the carrot. Likewise, avocados, which is the target the health function of the cervix of female, and it looks like the cervix and the organs, female organs, and it was traditionally claimed, but these days with the help of modern kind of molecular investigation, now it has been proved, and many patents are there for the avocados for the health function. Then this is the example of walnut, which looks like brain, and we know that now it has been reported for the investigated by many researchers for the purpose of the brain function. Kidney beans is one of the example which we consume frequently in India, like rajma and all that. And this rajma is having some phytoliths for the kidney protection. Then the tomatoes is traditionally claimed for the maintenance of the health of the homeostasis of the heart and cardiovascular system due to the lycopene and all that. There is many reports and patents on tomatoes. Then figs, which is having beneficial effects on sperm and sperm counts. Then mushrooms, which resembles the structure of our ear and we know that these days this is described for different type of secondary metabolite obtained from mushroom is for the human ear function. Then grapes is very beneficial for the different type of alveoli function. So likewise, different other function like GIT is function of GIT is maintained with the help of ginger. Then sweet potatoes, which looks like pancreas, and now it has been reported for the purpose of glycemic index and for the diabetic patient. Then different type of olive extract and preparations are function for the ovaries, and ginseng and ginseng pharmaceuticals are now for the purpose of stimulant and relaxant worldwide. Various kind of teas and various products are marketed. These are the some opportunities in India, like Harisingh, that is Parijat, aloe vera, giloy, ashwagandha, and many others which have huge scope for the research for the purpose of COVID-19 pandemic. So to conclude, ethnomedicinal inputs plays very important role in quality control and standardization processes, and modern evaluation and analytical procedures should be employed to explore and investigate traditional and ethnomedicinal experiences. Micro learning is one of the very important area of learning capabilities, and learning attitude can be developed with the help of micro learning. And of course, micro learning in herbal research facilitates holistic approach of investigation. Quality attributes like upside attributes for counsel must be dynamic in terms of their quantum and evaluation. Standardization may be considered as one of the effective tools for the quality control, and we know that this can be achieved by involving the integration of modern and advanced analytical techniques. So by this way, with the collaborative efforts, definitely the challenge of the phytomedicine could be achieved in big way for the purpose of the drug development from botanical and natural sources. So this is my email ID I have mentioned here in this slide, and if you ask some question here only, I can try. I'll answer here, or you can put your question through email also. Thank you so much.
Participants may ask their queries if they are willing to ask.
I have one question.
Yes, please.
Sir, if we have ethnomedicinal data with us, but a large number of ethnomedicinal data relating to various plants which are used for curing certain diseases. So how will we scientifically validate that particular plant is useful for treatment of a particular disease? That is, we are getting so many.
Yes, very good question, Shiv Sekhar. Your name is Shiv Shankar Shukla. What's your name?
Sir, I am Gunjan Kalyani.
Okay.
Gunjan Kalyani.
Okay. Oh, I'll talk to you. So this is very good question, and ethnomedicine input. Whenever we know huge number of list is available in terms of ethnobotany and ethnomedicinal claim. So when there is we pick one particular plant for the therapeutics and one particular bioactivity, our efforts must be starting with the collection of the plant material from that original site where it is reported. Say for example, if one plant is reported to be used in one region, maybe located in Chhattisgarh state, say for nearby Raipur or maybe Durga or any other places, then if report is available in Canada or a report is available in Delhi, it doesn't mean the work should be started dependent on the supplied material. When we go for the ethnomedicinal investigation, once we have to visit that particular place mandatorily where it is claimed, and on the basis of the material which is available in that particular geographical location, the purpose is to find out in what way the on the ground factors is contributing for the overall biosynthetic pathway. So altogether, whatever information is available in the text, what particular information is available at present at that location, and in what way the underground and underground factors are affecting the biosynthetic pathway of those plants. That will be collected in proper way, and ultimately the plant material should be collected from that site only. And it must be evaluated in terms of its wholesome quality. If we are able to develop metabolomic profile, then that will be the best way to develop the ethnomedicinal claim. So in this way, we can start with the plant material directly collected from the site where it was claimed for the first time. Is it clear?
Yes, sir. It is.
So there is a question from our YouTube participants also. So kindly consider it. I'm transferring it to you.
Yes, please.
What are the advantages of phytomedicine approaches?
What are the advantages
of phytomedicine approaches?
See, the question is advantages. There are many advantages of phytomedicinal investigation. The basic advantage of this medicinal investigation is to decide the safety and effectivity. See, plant may be claimed for many reasons, many different type of claims. How much plant is safe and how much it is effective for the purpose, that is the main target of such type of investigations.
Okay, sir. Thank you, sir. Thank you, sir, for sharing your deep knowledge with us. It will be a great pleasure for us to listen to you again in our future events. I once again thank you.
Thank you, madam. Thank you all, and I love to have some queries and remarks through email also. Sure. If someone is writing at least one line, I will be more than happy. I will think that my 1 hour was consumed for the betterment of the profession. So thank you very much, all which were involved directly and indirectly in the whole session, which was one of the part of this STTP organized at Columbia Institute of Pharmacy. Thank you very much.
Thank you, sir.
Thank you, sir.
So may I leave, Ruchi ma'am?
Yes, sir. Sure, sir.
Thank you.
For our third lecture session, kindly join at 1:20 p.m. by using the same link. I once again thank you all for your active participation.